Good afternoon, everybody. My name is Dwayne Moose, and today I'm going to talk about a computational circuit topology tool that I've been developing over the past 2 years while working at the MSBB lab. I will also discuss two of my articles that were published while using this computational tool. Massive amounts of effort went into development of the computational circuit topology tool. In the end, it was released and a complete tool is now currently available online for free. Before we go into the details of this computational tool, I will reiterate the basics of circuit topology. Circuit topology is based on defining pairwise relations between intramolecular contacts. One can distinguish three types of relations between two contacts: parallel, series, and cross. These contact arrangements are topologically distinct. Therefore, any transitions between them would involve the formation and rupture of contacts. Translating the complexity of protein structure in a functional and accurate topology framework allows us to understand the complex network of structural relationships and provide insights into folding mechanisms, conformational dynamics, and folding stability, which ultimately supports protein and drug design. The main goal of this project was to develop a tool that allows for external use and customization of the concept of circuit topology. Having as many people as possible using this tool and some even publishing research based on circuit topology is vital for the evolution and longevity of circuit topology. The tool in its current state allows a user to completely have a circuit topology workflow within one click of the button. You have the importing of protein structures, which will then be translated to circuit topology contact maps. These contacts are brought into a circuit topology matrix, and this information can then be utilized to do further circuit topology related analysis. To show how a user of a program can easily perform an analysis of basic properties and to demonstrate the efficiency of our algorithms used in the protein circuit topology software, we ran the code on all 23,000 entries of the AlphaFold database and mapped the universe of human proteins to a circuit topology space and performed a preliminary analysis of basic properties such as length, relative contact order, function, and cellular location of the proteins. Next, we provided an example of how various filters can be applied and examined the consequences on the circuit topology of a protein. The figure shows the results after applying filters that remove repulsive Thomas-Dill energy contacts and after filtering out contacts that consist of 2 residues within a distance of 25 residues. One can quickly notice the effect both methods have on the number of residues in contact. The length filtering method shows the biggest difference. The residue-residue contact matrices shows a large gap in the middle that is caused by exclusion of the short-range contacts. But more importantly, one can also see that the circuit topology matrix shows a relatively high number of cross contacts when compared to the previous methods. Here we demonstrate 3 of these filters used in parallel, but the reach of possibilities is far wider than currently shown. If needed, users are able to use the desired filter types in a desired order and chain them together in order to attain the demanded level of analysis. Macromolecular assemblies such as protein complexes and protein RNA condensates are involved in the most fundamental cellular processes. The arrangement of subunits within these nanoassemblies is critical for their biological function and is determined by the topology of physical contacts within and between the subunits forming the complex. Describing the spatial arrangement of these interactions is of central importance to understand their functional and stability consequences. We extended the functionality of the computational circuit topology tool to be able to define the topology of a complex consisting of linear polymeric chains with inter- and intra-chain interactions. We apply our method to a system of model polymer chains as well as protein assemblies. Our multi-chain circuit topology should aid analysis and predictions of mechanistic and evolutionary principles in the design of macromolecular assemblies. Both the circuit topology framework itself and the respective software were extended to include more types of contact relations concerning two or more chains. These new relations can be categorized in either looping contact relations, tandem, and independent relations. In this example, a yeast complex composed of 6 different proteins was analyzed using a computational tool, and a corresponding circuit topology matrix was created. The complex has 250 interprotein contacts and 958 intraprotein contacts. One can quickly notice the 6 distinct areas in the circuit topology matrix corresponding to the protein chains. We also developed an algorithm to determine the circuit topology of protein complexes and condensates. For this aim, we used the most comprehensive database containing stoichiometric and functional information about protein complexes, named the Complex Portal. From the Complex Portal, we received 117 protein complexes containing over 1200 proteins, and these then were used to obtain the stoichiometric information and to calculate the complex heterogeneity index. The heterogeneity index can be used as a measure of how many different proteins are inside the complex. The heterogeneity index is calculated by dividing the number of different proteins in the complex by the total numbers of proteins in the complex, where a score of 1 means that all proteins inside the complex are structurally different, and a low score means that there is a form of stoichiometric asymmetry present in the complex. The average topology fractions of the topological states for all protein complexes are shown in figure A. The topology independent 2 has the highest fraction compared with the other states. This is the result of the complexity of the structure and contacts inside each protein of the complex, which is in turn reflected in the rise of the independent contact pairs. The reason behind the analogy can be attributed to the compactness of the individual chains, which enhances the intra-chain contacts and the proximity of the chains inside the assembly, thus increasing the interchain contact probability. For the protein complex of our study, the average heterogeneity is 0.7. Figures C and D show the distributions of the topological fractions and the complex heterogeneity. As it is shown, the distribution of both the I2 and I4 topological fractions is highly localized at higher heterogeneity indexes. To investigate the circuit topology of a multi-chain system, we built a model composed of 100 chains in a simulation box. The system is initially equilibrated for a low cohesive strength and 0 bending rigidity. Then the simulation is started and the configuration of the system was sampled every other frame, which allowed us to compute the CT fractions during those frames. Figures A to D show the snapshots of the chain system having different cohesive strengths and bending rigidities, and the effects on the circuit topology fractions are shown below. To examine the effect of the isotropic to nematic transition on the circuit topology, we built a system with 900 chains, which are initially equilibrated in a cubic box of size 10. The size of the cubic box changes from 10 to 5. Figure C shows 2 snapshots of the system at isotropic and nematic phases. Figure D shows the looping contacts, which are monotonically increasing with the system concentration and the nematic transitions. This is due to the fact that the looping topology state can capture the local reordering of the chains at the isotropic nematic transition. In the end, we managed to successfully develop a computational circuit topology tool called ProteinCT. This tool allows for both visualization and topological analysis of protein molecules as well as protein complexes and condensates. The topological information can then be correlated to the physical and functional properties of proteins, which ultimately provides means of classifications of proteins. There are endless possibilities to serve as a valuable tool for a wide range of functional and evolutionary studies, and the download can be found in the link below. Thank you for listening. If you have any other questions related to the circuit topology tool or my research, you can reach me on the mail below.